Welcome to Lecture 3.3, in which we go over eukaryotic cells and organelles. This incredibly beautiful image here shows a single eukaryotic cell with various organelles stained in different colors and overlaid together. In this video, we will describe the general characteristics of eukaryotic cells, as well as their components and the functions of each component. We will also learn about the Endosymbiotic Theory. By the end of this video, you will hopefully also be able to compare and contrast prokaryotic and eukaryotic cells, and also animal and plant cells. Eukaryotic cells make up the entire domain of eukarya. This is a very diverse group that contains animals, plants, fungi, and protists. They can be unicellular or multicellular, and they are distinguished from prokaryotic cells by the presence of a nucleus that encloses the DNA. They also contain some other complex structures called organelles. Here we have representations of a typical animal cell on the left and a typical plant cell on the right. Like all cells, they have a plasma membrane, a cytoplasm, DNA, and ribosomes. We will cover the plasma membrane in more detail in the next lecture and discuss the other components one by one in this lecture. Let's start with the nucleus. The nucleus houses DNA, which contains all the genetic information of the cell. The DNA is represented by all these wavy purple strands. Unlike prokaryotes, which have a single strand of circular DNA, eukaryotes generally have multiple strands of linear DNA. When DNA is condensed, it forms a structure called chromatin. The nucleus is surrounded by the nuclear envelope, which is made of two plasma membranes that separate the inside of the nucleus from the rest of the cell. These orange structures are nuclear pores, which allow for transport between the cytoplasm and the nucleus. In the center of the nucleus is the nucleolus. This is where ribosomes, our protein-making machines, are assembled. The cytoplasm houses everything inside the cell, including the cytosol, the cytoskeleton, ribosomes, and organelles. The cytosol specifically refers to the gel-like fluid that makes up the cytoplasm and surrounds everything inside the cell. The cytoskeleton is a network of proteins that helps the cell maintain its structure. Side note, cyto means cell, so it's quite literally called the cell skeleton. Organelles refer to membrane-bound compartments within the cell, including the nucleus, and add complexity to the eukaryotic cells relative to prokaryotic ones. As mentioned, ribosomes are protein-making machines. They are themselves made out of proteins as well as some RNA. Ribosomes can either be free-floating in the cytoplasm or attached to the rough endoplasmic reticulum, as depicted here. We will talk more about ribosomes when we go over the process of translation. Like in prokaryotes, the cell wall is a structure that surrounds the plasma membrane and provides structural support and protection. It is generally found in plants and fungi, but not animal cells. It helps hold the cell together when water flows in to keep the cell from bursting. In plants, the cell wall is made of cellulose, and in fungi, the cell wall is made of chitin, both polysaccharides that we touched on in our previous lecture. Now let's start going through the endomembrane system. The endomembrane system is composed of several different organelles and parts of the cell, including the nucleus, the endoplasmic reticulums, the Golgi, the lysosomes, ribosomes, and the plasma membrane. These work together to make various proteins and lipids for the cell. The endoplasmic reticulum, abbreviated as ER, is a large network of membrane-bound sacs that are connected together as depicted here. The membrane is continuous with the nuclear envelope. The rough ER is the part that is covered with ribosomes, which gives it a rough texture. The smooth ER is the part without ribosomes. The internal space of the ER is referred to as the lumen. The rough ER is right next to the nucleus. Its main role is to perform protein synthesis through the ribosomes embedded on it, and it will also do some protein modification. You can think of it as a protein factory. The proteins made by the rough ER will eventually be delivered to various parts of the endomembrane system, and some of them will get secreted, which means that they enter the plasma membrane or leave the cell entirely. The smooth ER extends from the rough ER, and its role is to synthesize various carbohydrates and lipids such as steroid hormones. You can think of it as a lipid factory and carbohydrate factory. In the liver, the smooth ER also helps with detoxification. The Golgi is a collection of flattened stacks of membranes, which are called cisternae. Continuing our analogy of the ERs being factories, you can think of the Golgi as the post office of the cell. Once the proteins and lipids are made, they go to the Golgi to be sorted, packaged, and distributed. The macromolecules are delivered using vesicles, which are membrane-bound vessels used for transport within the cell, like a delivery truck. You can think of the word as being a combination of vessel and vehicle. The Golgi has two faces. The cis-Golgi is the side facing the rough ER. It's the part of the Golgi that's on the same side as the rough ER. The trans-Golgi is the side facing the plasma membrane, so it's the side that's on the opposite side as the rough ER. Over time, the cis-Golgi matures into the trans-Golgi. As this occurs, the proteins and lipids are further modified, usually with the addition of carbohydrates. In plants, the Golgi also helps produce polysaccharides for the cell wall. Lysosomes are the last organelle in the endomembrane system that we will discuss. They contain digestive proteins. If the ER is a factory and the Golgi is a post office, the lysosome is a garbage disposal and recycling center, breaking down things that are old and used so that the components can be used to make new things. Its acidic environment and some specialized proteins allows it to break down macromolecules and even worn-out organelles. It is also involved in endocytosis, a process of a cell internalizing something from its environment. Incidentally, lysosomes are only found in animal cells. Peroxisomes are small organelles that oxidize fatty acids and amino acids by adding oxygen onto them. These processes lead to the production of hydrogen peroxide, which they can then also break down, hence the name peroxisome. Vacuoles are essentially big vesicles that can serve to provide temporary storage space, perform digestion, and take up water. They are mostly found in plants and fungi, but can also be found in animal cells. Plants have a large central vacuole that can take up the majority of the space inside, as depicted in this illustration. Mitochondria are famously known as the powerhouse of the cell, due to their function in providing energy in the form of ATP. You can see that it has two membranes. We will go through its structure and its role in producing ATP in future lectures. Notice that mitochondria actually have their own set of DNA and ribosomes. However, they still rely on proteins provided by the rest of the cell. Chloroplasts are found only in plants and perform photosynthesis, and we will learn about its structure and role in more detail later as well. Like the mitochondria, the chloroplast also has two membranes and its own set of DNA and ribosomes. This brings us to the Endosymbiotic Theory. This idea was spearheaded by the evolutionary biologist Dr. Lynn Margulis, and proposes that mitochondria and chloroplasts descended from a prokaryotic cell that was initially engulfed by a eukaryotic cell, but then formed a symbiotic relationship with it. That is likely why the mitochondria and chloroplasts both have their own set of DNA and ribosomes. Some of the evidence for this theory includes that they divide independently from the rest of the cell, have circular DNA like prokaryotes, and if you recall from the scale of life slide in lecture 3.1, are a similar size to prokaryotes. Their double membrane may be a membrane that was formed around the original prokaryote when it was engulfed, as shown in this diagram. Finally, the cytoskeleton. As previously defined, the cytoskeleton is a network of structural proteins. These can be divided into three groups. The first is microfilaments, which are the smallest and are made out of proteins called actin. They help with transport within the cell movement, and muscle contraction. They are shown in red in this image, and you can see that while they are spread throughout the cell, they are brightest at the periphery of the cell. The second is microtubules, which are the biggest and made of proteins called tubulin. They also help with transport within the cell, and make up centrosomes, which play an important role in cell division. They are in green in this picture, and are present more towards the inside of the cell. Intermediate filaments are mid-sized, and provide structure for the cytoplasm and anchoring of organelles in the cytoplasm. They are the most durable of the three and do not break down easily. In summary, eukaryotes have linear DNA stored in the nucleus in the form of chromatin, as opposed to prokaryotes that have circular DNA in the nucleoid. Eukaryotes are also highly complex and compartmentalized into organelles, with many components that each have their own function. Some of these components are not found in all eukaryotic cells. For example, cell walls and chloroplasts are both found in plant cells, but not animal cells, while lysosomes are found in animal cells, but not plant cells. The mitochondria and chloroplast are a bit special because they have their own DNA and ribosomes, and this is because they likely descended from a prokaryote that was engulfed by a eukaryotic cell a long time ago. The prokaryotic cell and the eukaryotic cell had a symbiotic relationship, and eventually this led to the mitochondria and chloroplast. This is the end of the video. Thanks for watching.